Thanks for joining us. I'm your host, Raquel Faria. I am an internal medicine consultant at the Centro Hospitalar Universitário de Santo António in Porto, Portugal. Today, I'm joined by Professors David Isenberg and Ricard Cervera from the Lupus Academy Steering Committee. Between them, they will be providing expert insights from the key research and clinical data presented at this year's EULAR 2024 Congress that was held in Vienna in June 2024. David, Ricard, welcome, and thank you so much for joining us today.
Thank you, Raquel. Thank you.
Thank you. Greetings from Barcelona.
Thank you. We saw several reports of clinical trials assessing novel therapies in SLE during the Congress, but before discussing those, I would like to start by your global insights of the meeting. David, what did you think about the global meeting?
Well, I enjoyed the meeting a lot, actually, and that had partly to do, to be honest with you, with the geography of the centre, because the previous year in Milan was complicated because it was spread over various floors. This was all on one level. It was nice and easy. I didn't get lost this year, which was true. But I always say, why do we go to meetings like EULAR and ACR? And that's very simple. You go for two reasons. You go to learn and you go to get fit. I must have about 1,000 steps every day while I was there.
Ricard, what are your comments on the meeting?
Yeah, I agree with these comments from David. Certainly, I think that it was a big Congress because usually EULAR is a very big Congress with thousands of participants, but also it was well organized as usual. And I think that this meeting is also, let's say, something like a meeting point for rheumatologists and specialists on autoimmune diseases, where we gather with other colleagues, we discuss, we usually do meetings of the different task forces of EULAR, the different steering committees, ad boards. So it's also a meeting point for many people, in addition, obviously, to all the classical scientific presentations, oral presentations, plenary sessions, and so on. And for sure, it's a place that we have to walk a lot, because certainly the distances from one side to the other are really very, very long. And well, this is also important because we do a sport while being there, so that's good.
And I like also to comment something that I think it was the first time in EULAR they organized an escape room on rheumatology, and I applied for that with a group of different colleagues from all over the world that we didn't meet, and it was really amazing. And I think it was a wonderful initiative also. Talking about lupus, the first How to Treat session, that is the classical conference plenary session on the EULAR, was on lupus, what's on the horizon, and it was presented by Laurent, and we both moderated it. David, what do you think about the perspective that Laurent transmitted on what's on the horizon for lupus?
So for me, Laurent's talk was what I would call a 360-degree talk. He covered everything. He looked at causation, he looked at clinical presentations, he looked at new treatments on the horizon. And I think if you just wandered in from the planet Mars and you knew nothing about lupus, at the end of that hour, you would know a lot about lupus, actually. And he has very powerful slides, very colorful slides, lots of action-packed slides to see, and lots of data in the slides. So I felt it was a very 360-degree kind of talk, looking at many aspects of lupus, actually.
Ricard, do you have any additional comments on Laura's presentation?
Oh, I agree, really. Laurent is a very good speakers with very beautiful and impressive slides. And I think probably the optimistic ideas, especially we can envision a cure for lupus in the next few years. Probably we will talk later on the new therapies that probably can give us a hope for a potential cure in the horizon.
Thank you so much. Now going to the abstracts revision and comments, and we chose to start on pros and patients' work. And the first one I would like your comments on was an abstract from Lupus Europe about the Lupus 20 insights. Lupus 20 was launched in EULAR 2023, so 1 year ago, to ensure that there was a global online way for patients to have their questions asked on lupus. And they did a survey on how many people entered the site, what were the questions that were more chosen to be answered. And actually, I was a little bit surprised about the 10 questions that were more followed. Mainly, and I would like your comment on that, mainly because some of them, I do think that probably the physician and the nurse would address with the patient. And some others, I don't know if there is a frequently comment on that. So please, Ricard, can you please start commenting on what do you think about these 10 questions that were mainly chosen by the people that went to the website?
Well, first of all, I have to say that I am very proud of this work done by Lupus Europe. I am one of the members of the medical advisory board of Lupus Europe, and this has been an impressive project that was started in France by the French colleagues and then was translated into English and then to several languages. I personally participated in the English, in the Catalan, and in the Spanish versions, and really I believe that this is a unique tool for patients, for the education of patients, and we have to spread this. I think that patients need good internet material for education, and this is really a very good material for education for them. It's certainly a little bit strange that 10 more visited questions, and some of them probably are the most visited because they were the first in the list. But probably this is also a challenge for us as physicians and as advisors for Lupus Europe, that we have probably to highlight more other questions that we really believe that are very important, that patients are educated in other questions. For instance, we have to emphasize on the importance of the different treatments, the adherence to the treatment, the potential side effects and the prevention of some side effects, vaccination, and other aspects that are really lifestyle aspects related to lupus, that it's very, very, very important that we educate patients on this. So probably the consequence or the lesson that we physicians have to learn is that we have probably to highlight some questions, to do something in order to make probably more popular many other questions that have not been selected by patients, and that we really believe that it's important that patients know. So really, I think that it is a good job, and more work is still needed in order to reach our goals with this project.
David, do you have additional comments on these 10 highlighted?
Only a sort of general comment, really, because I would generally support what Ricard said. I remember when I just became a consultant, I did a very simple thing. I asked 100 successively attending lupus patients at the clinic, what's the number one thing that troubles you most about your lupus? And I assumed it would be fear of death, fear of kidney failure, lipodialysis, and it wasn't. It was fear of fatigue, or troubled by fatigue. And it was the big thing that so many patients then and still now actually continue to complain of. And it made me think that clearly what troubles patients and what worries patients are not necessarily the same things as worry the physicians who are looking after the patients. And Ricard is right. We have to get our balance really in terms of what we tell our patients. We have to expect that patients are going to be worried about things which we wouldn't necessarily be thinking about, but for them, it's very, very important. And I think we have to keep wide-angle lenses open to accept any kind of question. But then, as Ricard says, we also have to try to guide some of those questions to really address some of the very important issues around drugs, side effects, vaccination. These are all very important questions.
And you just entered a little bit the discussion of the next abstract that was the result of the real world BioLag biologics registry that I'm absolutely sure you are involved in. And this abstract highlights that after rituximab, the response to treatment is discordant of how the patient feels.
Yeah.
So I'm going to ask first, Ricard, what do you think about this? This is not a new thing, but it highlights that patients also do, with rituximab specific, that is also this. So Ricard, what do you think about this result, and how do you feel it on your clinical practice?
Well, certainly these results stresses what David mentioned before, that there is a discordance between what we physicians believe is the most important issue, that in this case is the response to the inflammatory manifestations and so on, and what patients really believe that it's the most important for them, that are what now we call the type B manifestations, these fatigue, exam pains, brain fog, depression, and so on. And the terrible problem is that nowadays that we are having beta therapies, especially for these inflammatory manifestations, we don't have yet any treatment, good treatment, any drug for this type B manifestation. So probably patients will believe or will feel still depressed because they don't have answer to their main problems. We don't have good therapies for fatigue, for exam pains, for brain fog. And this probably is our challenge, is that we have to look for therapies for this. And also for the drug companies, we have to look or to ask drug companies to have better drugs, better therapies for this, and probably to include also in the tools in order to assess the response of the different treatments, also the response to this or the amelioration of these symptoms, not just to decrease in the joint involvement, the cutaneous manifestations, the renal features. We also must put in the outcomes the decrease of fatigue, the decrease of the brain fog, and a good treatment will be one treatment that also decreases this type B manifestation. So really, this will be a great challenge for the next years.
Thank you, Ricard. David,
so
this was particularly on moderate to severe SLE, which adds an additional challenge. So please do comment that.
So there are two things I'd like to highlight here. One is that when a patient comes back to see me after a major therapy, whether it's rituximab or cyclophosphamide, obviously I'm going to look at the DNA antibody levels, I'm going to look at the C3 levels. And I frequently, and I'm sure both of you have had this experience too, I'm always happy to say to the patient, great news, your DNA binding is coming down, your C3 is going up, and the patient, yes, but my joint still hurts, I'm still fatigued, I'm still not well. And so that's one part of this. Secondly, it is a question of time to response of certain symptoms. It's differentiated in rituximab in particular. So if I treat a patient with, let's say, hemolytic anemia or thrombocytopenia, I can expect to see improvements post-rituximab after 1 or 2 weeks. But if it's joint problems or skin problems, it could be 1 or 2 months. And if it's kidney disease, as we and Liz Lightson have shown, it could be 3, 4, 5 months. So there's a differential. And you have to say to patients, I know you're still not feeling well, but it's only 2 weeks since you had the treatment, or a month since you had the treatment. I'm still confident. I always try to be confident that in a few weeks you will feel a lot better. And this is particularly true when it comes to our new friend Benlysta, because it's very important. In fact, Rich Fury taught me this actually. Rich Fury, for those who are listening, is one of our fellow members on the board, and Rich was one of the first American rheumatologist to get very easy access to Benlysta. And he said to me, when I give it to a patient, I say to them, it could be many months before you begin to feel better. So, I'm going to put you on Benlysta, but if I invite you back in a month, I'm telling you, you're not going to feel any different. 2 months, maybe no different. After 3 to 4 months, you might begin to feel a bit better. So there is that time lag with biologic effect, which you have to express to patients. And so I think there are a number of reasons why these results were as they were. But it's clear there's not a simple relationship between you get a drug with these possible steroids, actually, of course, because steroids are fast acting, but they just have these side effects. But in the main, for drugs like cyclophosphamide, rituximab, Benlysta, it's going to be a while, several weeks, several months before you begin to see the improvements. And in a sense, when I give patients these treatments, I do say to them, don't expect this is going to wake up the next morning and feel great, because you're going to be disappointed.
Thank you. And there's a good time to jump to some abstracts about drugs that are being studied for lupus, but are not quite on the practice for lupus. So we chose to highlight deucravacitinib, so a tyrosine kinase 2 inhibitor that is an oral drug. So it's supposed to be taken every day. And 3 of these abstracts that we highlighted, 2 were oral presentations, 1 was a poster, and they are all sub-analyses of the phase 2 trials. One was on the evidence of improvement in skin and more sustained response in the drug arm. Other was on separate responses of different scores, global scores like ASRL4, BICLA, and also CLASI 50 and the joint 50. And it showed that not all together they had an effective response, but if they were single used, there was also an effective response on the harm versus placebo scores. And the last one was on the LDAS achievement on the study. And it looked like it was early and more sustained on the treatment arm versus placebo. So David, what are your global thoughts on deucravacitinib and these results?
Okay, so I think before I get to specific, just a little bit of background. So it's very important to remember this is a phase 2 trial.
Yes.
And as I think everybody listening to this podcast will know, there is unfortunately an increasingly long history of drugs like eprituzumab, ustekinumab, baricitinib, regerimod, looking good at phase 2 and then failing in phase 3. So I think we have to be a little bit cautious in our optimism as to whether this drug is going to make it. The results that we've seen so far are encouraging. We've seen some good responses on skin disease, and that's important. And it's very clear that there is clearly a gap in the market, because I'm sure it's the same for Ricard too. For me, the greatest frustration I have in my regular week is that on a Friday afternoon, I do a general clinic. I see patients with rheumatoid, psoriatic arthritis, ankylosing spondylitis, and I know that if the conventional drugs, methotrexate, sulfasalazine, hydroxychloroquine, if they don't work, it doesn't matter too much, because I've got at least five different types of small molecules and biologic drugs which are effective, which work often fairly quickly and are affordable, 5. And so it's almost impossible these days to find one or other of these 5 approaches, whether it's B-cell depletion or blocking the B-T-cell interaction or blocking IL-6, whatever. You can find something to help the patient. And then I go to the same clinic room on a Monday morning and I see my lupus patients. And now I can give them rituximab, that's pretty widely available. I can give them Benlysta in restricted conditions, and that's it. In the UK, we don't get to use anifrolumab. It's not been approved by NICE, so I can't use it. So there is clearly a gap because there are definitely patients who just don't respond to conventional drugs, don't respond particularly well to rituximab or Benlysta. And so if this drug meets its endpoint in phase 3, then there definitely will be a space for it in the market, provided it's not too expensive, because that's where it all gets very difficult. Voclosporin in the States costs over $90,000 per patient. I mean, that's unbelievable. So they've had to knock the price right down to be approved by NICE, but obviously it's still not as widely available as well, perhaps as manufacturers would like it to be. So these are the problems that I see with it.
Ricard, do you want to comment the results?
Yeah, I fully agree with David. Really, these phase 2 trials are generating great expectations, but we have to wait for the phase 3 because of the terrible bad history that we have with many other drugs in lupus in phase 3. But hopefully these signals will mean that phase 3 trial will succeed. And one important issue is that most of these sub-analyses that have been performed in these phase 2 trials are using clinical definitions that probably are more linked to our clinical practice. So it's low lupus disease activity or class C definition, not just the SRI, BICLA, that for many clinicians, it's very difficult to understand. If you say that patient reach SRI 4, means probably nothing for many clinicians, but if you say that patients achieve low lupus disease activity or remission, this is probably much more easy to understand because these are clinical endpoints. So I think that these are also signals that this drug will work. So let's hope that phase 3 will succeed. We had another small molecule that failed, that baricitinib, that we had great expectations, but hopefully this will succeed. The additional problem, in addition to these difficulties in demonstrated efficacy, the cost, and so on, is also the nomenclature, the name. Every time the name is much more difficult to pronounce. So maybe for English-speaking people it's easier, but for many other non-English-speaking people, really, to pronounce deucravacitinib, it's really difficult. So also let's hope that the drug companies will put an easier name for the drug at the end of the study.
Thank you. Now, one of the late breaking abstracts that we selected was in a drug that I really want to have your comments on. That was the safety and efficacy of daratumumab in systemic lupus. This was a single center phase 2 open label trial. But the thing is, the daratumumab is an anti-CD38 monoclonal antibodies, which is a bigger depleter of B cell types. And actually there were good results in these 10 patients where it was done. And it was a very quick and long response. And these were all refractory cases. So David, what's your insight in daratumumab, and which patients you do think it might be considered?
I think the last question is still sadly unanswerable. With these things, you feel that there's a little something of being dangled in front of you. This
Yes.
And you're going to reach it and you can never quite get it. And I think with these drugs, it's much so. I remember when eprotuzumab reported, looked fantastic. Anti-CD22, it seemed to make biologic sense. Everyone was very excited, and then it failed in phase 3. And so this is, as you say, small number of patients, okay, resistant patient, which makes it a little bit more interesting. It reduced DNA binding, so it's clearly having a genuine biologic effect. So I'm always, as I say, cautiously optimistic in this situation that this will be a real phenomenon. But I'm afraid, from bitter experience, we now know that until 1 or preferably 2 phase 3 trials have been shown to be successful, and preferably in renal and non-renal lupus, I wouldn't get too excited. I would be a little bit excited.
Okay, Ricard, do you want to comment anything on daratumumab, which is a little bit easier than deucravacitinib, but not quite easy yet?
Yes, that's right. A little bit easier, but not too much. Well, the good thing is that it targets probably one of the cells that we really believe that are clearly involved in the autoantibody production in lupus and persistence of the disease, which is the long-lived plasma cells. And I have to tell you that we have also personal experience with this drug and in really refractory cases, and it has been the solution so far for these refractory cases. So I would say probably a similar word. So we have optimistic expectations, but let's wait for additional data. But so far, so good.
There's something about these 2 drugs that we are importing them from other diseases. So deucravacitinib is being used on psoriasis, and daratumumab is being used for plasma cells, neoplasm. So as historically we've been doing in SLE, these 2 drugs were imported. So even if we want the clinical trials to have a result for them to be used on label, but in difficult cases, sometimes we need to import them and using these, but this is going to be case by case always. There is a different story with other drugs that are developed for SLE, as belimumab was, and there is actually an oral presentation abstract that we wanted also to highlight, which is the phase 2 safety and efficacy of subcutaneous ianalumab. And ianalumab is a novel monoclonal antibody targeting BAFF, like the BAFF receptor, like the same way as belimumab, but this antibody has actually cytotoxicity, antibody dependent, that would mediate the depletion of B cells. So this was a longer one than the previous phase 2. So the patients were followed until the 52 weeks, and it looked very well tolerated. Ricard, do you have any insights on this drug? We have been hearing it for phase 2 for several congresses now and publications. What do you think it's going to be of an added value if it goes further?
Yes. Well, first of all, I would like just to comment on what you said just at the beginning of this presentation of this abstract, is that fortunately we had some drugs that have been approved for other conditions and that we are taking for lupus, and this has been the success of most of the drugs, certainly, that we are using, and we have improved our patients' behavior thanks to these drugs coming from other diseases. And unfortunately, those drugs that have been just developed for lupus and they are not in the market, probably some of those that fail in the trials probably were as good as these others, but as we cannot use off-label, we have never been able to demonstrate the efficacy. And in this case, this one is similar to belimumab is of the same co-stimulation pathway. So we can expect also good efficacy in patients with lupus, but the only information that we have is this from these trials, because it is not in the market for other conditions. And so far the information is quite interesting. I would also like probably to remark that in this, at least in this study, in these abstracts, they have included in the as an endpoint the composite endpoint that includes SRI plus a corticosteroid responder criteria. That is something that we are now emphasizing, is that if we need these new drugs, one of the main reasons is in order to avoid steroids use, because steroids actually are a very good drug to decrease inflammation. The problem is that they produce side effects. So one of the ways to demonstrate efficacy of a new drug is that you can reduce or even stop, completely withdraw the use of steroids. So in this abstract, they specifically include this corticosteroid responder criteria, which I think that gives enough importance to the good results of this abstract.
Thank you. David, do you have additional comments on ianalumab?
So first, just a general one. I always say when I talk to the pharma company representatives that there's a kind of, for me, a sort of what I call a holy trinity effect when it comes to analyzing the potential value of a new drug in lupus or indeed any disease. Number one, does it work? Number two, what are the side effects? Number three, how much does it cost? And you can't get away from it. You could have something which is very successful, but its side effects are unacceptable. The price is also unacceptable, and it's not going to fly. You could have something which is sort of okay, but the side effects again are just not so good. Or you could have something which it's kind of okay. The side effects are not too bad, but again, the cost is just too much. So you've got to satisfy these three things. So with this drug, it's looking good in the sense that the results are encouraging. This is some very nice safety data based over a year. Again, it's clearly doing good things for the patients who have it. But again, it's in phase 2. We're looking for phase 3. So again, my enthusiasm is tempered. When I see great results in phase 3, then I get excited.
Thank you. Let's move to another category. And one of the abstracts that we highlighted was the longitudinal assessment of biomarkers in nobility, the phase 2 clinical trial of obinutuzumab in proliferative lupus nephritis. And actually, this work led by Ed Vital looked at the peripheral B cells, B cell subsets, and serum biomarkers in these patients. And actually, by week 4, if the response had reduced B cells in these patients compared to the placebo, there was a longer sustained period of response in kidney. So, Ricard, what are your thoughts on this impact?
Well, certainly we are just waiting for the results of the phase 3 trial with obinutuzumab, the Regency trial, and while waiting to have this additional data from the phase 2, from the mobility study, I think that increases our great expectations of obinutuzumab. Actually, I think that we all believe that targeting CD20 like rituximab is really very good, and we all are expecting that finally a proper trial, phase 3 trial, demonstrate efficacy. So I think that we have great expectations with obinutuzumab in phase 3. While waiting for these phase 3 results, these data, additional data on the phase 2 nobility, I think that are really very good. It really gives us more good expectations on the success of the phase 3. But obviously we have to wait for the final phase 3 data.
David, this is a phase 2 and they're waiting for the phase 3, as Ricard just highlighted. But actually, we do feel this is going to give some justice to the effect of rituximab in SLE. So we are all waiting, almost like this is going to happen. So what do you think of this?
So I think there's a couple of elements to think about here. So why might this be a very useful drug? Well, it's quite simple, really. Rituximab, I think most people would accept that in spite of the 2 failed trials about 13, 14 years ago, for many patients, it has proven to be a very useful drug. It's now very cheap, and it's effective. The problem is that it's 20% mouse protein, because it started off as a mouse protein. It was converted by the scientists who worked with it into human, but 20% mouse protein. The consequence of that is that about 15%, maybe as high as 20% of patients given this drug get an allergic response to it. And I now have a group, I've now treated just over 200 lupus patients with rituximab, and I have a group of about 20, 25 patients for whom it clearly worked, but I can't give it to them because they're allergic to it. So the notion that you can now use a fully humanized anti-CD20, which is what this is, to replace it is extremely exciting. The results of the trials in phase 2 were okay. I didn't think they were sensational, actually. You had to wait quite a long time until you saw the difference between the placebo-treated and the drug-treated patients, trial-treated patients. So I remain quietly confident that it's going to work, but I want to see the data, just like Ricard. I want to see it.
And talking of CD20, there was also an abstract of real-world clinical practice from the Lupus registry that showed the effectiveness and safety of rituximab in lupus nephritis. And actually, this is a result. So it worked on the real-world data, as we are all aware. So given your extensive experience, David, with rituximab, these kind of results of abstracts, how do you think this impact the clinical use of rituximab?
So I'm sorry, one thing I forgot to say, actually Ed Vital's work for his PhD and subsequent studies is a very important thing, because what he shows is, as Ricard touched on, the more complete the B-cell depletion, the longer the remissions tend to last. Well, not in every single case, but as a general rule, that's the way that it seems to work. So what we're getting here is what I call real-world data. You're seeing patients who've had rituximab in a number of different sort of clinical situations, but especially in lupus nephritis, which, again, for a number of patients remains a problem. I confess that I quite like using drugs like rituximab and cyclophosphamide, especially for the more severely ill patients, because I know that they've actually had it. The studies which we've discussed in hydroxychloroquine show that, unfortunately, many lupus patients don't actually take the drug either at all, or sometimes they just take it in an inadequate dose. With rituximab, cyclophosphamide, it's no question. They turned up, they had it, or they didn't. You know, you know that. So that's why I found it quite useful. So although, of course, from the patient's perspective, I can understand why an oral drug, such as we've been discussing earlier in this discussion, is kind of nice. But actually, where you've got patients, and sadly I have several, and I'm sure you both have too, where you're pretty sure the patients are not very reliable tablet takers, to have drugs like these B-cell depleting drugs, cyclophosphamide, it's actually a good thing.
Moving on to real world also use of rituximab on one of the most unmet needs in lupus currently, which is neuropsychiatric. From the BILAG British registry, an abstract oral presentation was presented, indicating that rituximab was effective to treat moderate to severe NPSLE with lower steroids dose at 6 months. So, David, you have experience with rituximab in neuropsychiatric. What do you think of these kinds of results?
Okay, so there's a very important caveat about this study, which I'll come to in a second. But I think I do want to highlight the fact that every single lupus trial, every single one, excludes CNS lupus effectively.
Yes.
Every single one. So we're left in this very challenging situation where a significant number of patients have these wide varieties of CNS features, and yet we have no real knowledge as to whether or not B cell depletion or indeed Benlysta or indeed anything else actually works or might work for those patients. So it's very frustrating, I find. The challenging thing about this study is that a very significant number of patients said to have CNS lupus was said to have CNS lupus on the basis of a lupus headache. And that does give me cause for concern, because I'm not a great believer in the frequency of the lupus headache. I accept the fact that some patients will come into the category, but it's quite a large number of patients in this group were diagnosed with having CNS lupus on the basis of having a headache, and that does give me some cause for concern. That said, I have treated some of our lupus psychotic patients with this treatment. I've treated patients with a whole variety of other CNS features, and yes, I've certainly seen successes, but these are a small number of individual cases. We lack any clinical trial data. We rely upon studies like this one to give us some inkling, some notion that this might be a successful way to go, but it's deeply unsatisfactory.
Ricard, do you want to comment?
I agree with these comments, especially with the fact that really the severe, clear neuropsychiatric manifestation of SLE are rare, but really are terrible, and we need therapies for this, and we all have the good experience with rituximab. So if you have this real-world data that confirms and gives enough food for using rituximab in these severe neuropsychiatric manifestations, I agree that most of the neurological, psychological, neuropsychiatric manifestations in lupus are mild, are headache, anxiety, depression, and other manifestations that really don't require immunosuppressive drugs. But for those severe transverse myelitis, psychosis, polyradiculoneuritis, severe chorea, and all these, really we need therapies, and unfortunately we don't have these data from the trials, because are always excluded. We need data from these case series or from these cohort studies and registries. So good news.
Thank you. Actually, it's a little bit unethical to do clinical trials, RCT clinical trials on severe neuropsychologic SLE. I think this is a big question that would take us here discussing for a couple of times. Let's move on to a group of abstracts that we just chose to highlight the glucocorticoid steppering and immunosuppressive steppering. So there was an abstract that I think we can comment, because we always discuss this, on the different definition of LLDAS by the Asian group. And they compared the outcomes of the patients on more than 2,000 patients that were followed more than 2 years of follow-up. And they compared the risk for mortality and damage accrual between having the LLDAS with 7.5 steroids and 5-point steroids. And actually what they found was there was not a difference between the outcomes that they measured if the LLDAS definition had 7.5 or 5. So the hazard risk for the damage, for example, was 0.47 and 0.48. So no statistical difference between them two. And so they concluded that there was no evidence to use another glucocorticoid threshold for the LLDAS definition. So, Ricard, what do you think of this conclusion? What do you think this should change a little bit the way we see our targets?
Well, I think that these data will be controversial, because other studies have shown that certainly there is difference between 5 and 7.5. And the reason is that there is always a balance between you know that certainly 7.5 prevents more relapses than 5, but certainly also produces more side effects than damage than 5. So this balance between the prevention of flares and the production of side effects is what is really difficult to demonstrate. And maybe one study probably focused more data on the prevention of damage and another on the prevention of flares. And depending on the extension of the study, probably you can see more information on the prevention of flares or on the production of damage. So I think that there will be a lot of controversy. It will be very difficult to say that it's better to stay at 7.5 or 5. And I think that the good common sense dictates that if you are able to decrease to 5 or even to 2.5, is certainly better than if you keep on 7.5. But the data is the data. So I think that they have done this. This group has a large experience on. They define the low lupus disease activity scale. So I think that it is a data to keep into consideration. But this will remain obviously controversial. Let's keep for now that low lupus disease activity is 7.5 milligrams prednisone, and let's keep for now that the EULAR recommendations are to be below 5.
Yes.
Let's have both concepts on the table, on the floor.
Thank you, Ricard. David, I will ask you to comment with additional comment in another abstract that, of course, we all remember that 2 or 3 years ago, the French groups presented in EULAR that when 5 milligrams was stopped of prednisolone on patients that were in clinical remission, the patients tend to flare more. And then this year in EULAR, another group presented that it was safe to withdraw and to taper the steroids and discontinuing after tapering, even after tapering from 5. And this was associated with a low risk of flare. And this was an European study also. So can you please comment on this, that data are always searching with?
So a couple of things to say. The notion that there's not much of between 5 and 7.5, what's 2.5 little small milligrams sounds nothing, but actually Michel Petri shows very clearly that with every milligram above 5 milligrams that you go more damage over time. So I think the emphasis has to be, in spite of this abstract, on trying to get the steroid dose down. And I think Eric Morant, who put this paper together with his other colleagues, wouldn't actually deny that, actually. So for me, to get the dose as low as possible is important. The difference between these two abstracts, the French study, and my experiences would accord more with the experience from Paris, actually. It may relate, I don't know if it does, but that's because we're not told the data, to the serological state of the patient. Because for me, there's a difference between a patient who is deemed to be clinically inactive but is serologically not inactive, or in other words, active. So a patient who has high DNA binding, low C3, but clinically seems to be fine, is, I think, different from a patient who is serologically normal and clinically inactive. And it may be, I don't know, but it may be that in Paris, these patients who are on 5 were more serologically active and therefore more likely to flare. That's a possible suggestion which could be looked into relatively easily. So I think these are the two elements. I'm a great believer in cutting the steroids as low as you can do, but accepting, I think it is true, there are certain patients, quite a few actually, where 5 milligrams seems to ward off the evil lupine spirits, keeps them away. It's not a high dose. It's a pretty safe dose according to Michel's work. So I personally, especially if the serologies are abnormal, tend to keep those patients on steroids until the serologies normalize.
There were a lot of other abstracts on tapering, on lupus nephritis, on an Asian big group that highlighted that tapering, steroids and immunosuppression could really increase accrual of damage through time. But I do think there are different realities. And we want to, on these next few minutes, to finish the podcast, to discuss on CAR-T cell treatment on rheumatoid diseases. So we highlighted 3 abstracts on CAR-T cell treatment. 2 of them are on autoimmune refractory disease. One of them has actually a mean 18 months of follow-up, and other was on the Castle phase 1 and 2 trials, and another one specific on lupus nephritis. So, David.
Okay, so this was the hot topic. Wherever I was at this meeting, CAR T cells in the scleroderma session, the myositis session, the lupus session, it's everywhere. I couldn't get away from it. But actually, I have to say, what I was saying before about clinical trials, I want you to completely ignore now. I'm going to try and persuade you about something different, because I think there is a fundamental difference here because of even the results of a small number of patients. And historically, medically historically, it reminded me of the discovery of penicillin. So it is a fascinating story. So it was identified by accident in about 1928-29 at St Mary's Hospital in London by Alexander Fleming. But the real hero for me was not Fleming, although he made a very good observation, but rather a guy called Ernst Chain, who was a German Jewish refugee. He got to London, and in the war years, the early part of the war years, he actually synthesized and was responsible for the increased production of penicillin to the point where you could give it to patients. And he treated 6 patients. That's all he did, 6 patients. And the difference in the results from those patients was stunning. These were 6 patients with very bad sepsis, all expected to die. The first one was a policeman. And he did die, but he showed signs of recovery, which were regarded as very, very unusual. And the next 5 patients all survived. That's the trial of penicillin. That's it. That persuaded the UK and US governments to start mass producing penicillin. It was amazing. So you take this work, which actually relates to the work done by Georg Schett in Erlangen, and it's absolutely amazing to me anyway. So Schett actually has a nice sense of humor. I interviewed him at the ACR year before last, and he said, well, really, CAR T cells, it's like an expensive form of B-cell depletion is the way that he put it to me. And he's right, because at 350,000 euros per patient, it's very expensive. But the difference is stunning. I mean, after 12 weeks, in many of his 8 patients that he's described and published on now, these patients with DNA binding in the thousands have normalized. C3 very low, it's normalized. He gets them off all lupus drugs by 12 weeks. I never did that with rituximab. It's incredible. And then what comes back when these patients are followed up is a seemingly normal immune system. They have no autoantibodies. This is amazing to me. And the first patient was treated 3 years ago, and it remains completely well. Now, you could say to me, well, in a year or two's time, they could flare again. It's possible. But even so, the notion that you could get a very sick lupus patient off all treatment within 12 weeks, and that they're going to remain there with normalized responses to things like tetanus and to measles is to me quite stunning. There are caveats in the sense that he clearly chose his patients very carefully. They're quite young, they're very aggressive. These are not the patients that I see, plenty of who've had their disease for 15, 20 years, who have got some damage, and they're never going to go back completely to normal. But for me, it's a stunning observation. It needs to be confirmed, and there are at least 6 companies now trying to do that. And we saw some evidence of that at EULAR. But I think this is a hot ticket number.
Thank you, David. Ricard, do you already have experience with CAR T cell therapies in your patients?
In patients with autoimmune diseases, not yet, but our hospital has the first and so far the only academic CAR T approved by the European Medical Agency for patients with hematologic malignancies. And one of the problems of this therapy, that it's the cost, is much better than if you have to use a CAR T from a drug company. So in our hospital, they do this every week a lot, and extremely good experience. And even now we have the new dual BCMA CD19 that we have even great expectations. So we are now starting some studies on animal models also to apply this to lupus mice. So we really hope, as David, that this will be the future, will be probably the cure, but we have to select really the type of patients. And so far the information is for these very restricted, very severe cases that are really, fortunately, a minority. We don't know what will happen in those patients that have a disease less severe with a lot of B manifestations, type B manifestations. We don't know what is going to happen with patients that have more organ damage because of chronic disease. Also, we need to have probably long-term data, because now we know that some of the patients, a minority, but some of the patients that have received CAR-T therapy for malignancies develop a lymphoma several years later. So we need additional information, but certainly have great expectations for sure.
Ricard, do you want to have anything from the EULAR Congress that we didn't highlight during this discussion?
Well, just again to stress that we are probably facing a new area, on the management of lupus and other autoimmune diseases. And hopefully, let's see that in the next Congress in Barcelona, in the next EULAR Congress, we will see some phase 3 results, positive results of the trials that we have mentioned today.
David, despite your enthusiasm with CAR-T cell therapy, and you didn't use the word cure, but then Ricard used it a little bit slight. So what do you want to highlight from the old?
Yes, I think of it as the C word. I've always been very, very shy about the C word in lupus, but I think Ricard may be right. Let's hope for some patients at least it could be the answer. I think we didn't have time to discuss it tonight, but I think the idea of molecular signatures to try to distinguish subgroups of patients is interesting. There was a paper which we've discussed in the past, which seems to indicate there were perhaps 4 different groups. Judith James in Oklahoma has published perhaps 8 different groups, and at some point, the group that published at EULAR and Judy James should get their information together and see if they can combine. Maybe 6 is the right answer.
Thank you so much, David and Ricard, for joining me today and sharing your insights. To our listeners, we hope these discussions have helped you gain more of an understanding of the latest developments in lupus. Thank you so much for listening. We hope you enjoyed this podcast. Please leave us a review, which will really help us in reaching as many of your colleagues as possible around the world.